2024 Cross-disciplinary seminar series on metabolites hosted by the Office of Integrative Medicine and the Department of Biochemistry and Molecular Medicine. It's my great pleasure to introduce Dr. Jiyoung Lee, an assistant professor of biochemistry and molecular medicine at the School of Medicine and Health Sciences here at GW and a member of the GW Cancer Center. Dr. Lee received her PhD from the University of Illinois in molecular and integrative physiology before moving to the University of Chicago for her postdoctoral training in the Ben May Department of Cancer Research. During her postdoctoral training, she became an expert in cancer biology and made the novel discovery that mitochondrial metabolism can be targeted through a heme-binding transcription factor to sensitize breast cancer. Dr. Lee joined GW in 2019 as an assistant professor, and her lab has been interested in understanding altered metabolic signatures during cancer growth and metastasis, and its underlying signaling network using multiple cancer models, including breast cancer, and today we'll hear about head and neck cancer. Her lab has been funded from multiple sources, including the National Institute of Health and Department of Defense. She's been very active in training trainees of multiple levels, from masters to predoctoral and postdoctoral levels. So today, Dr. Lee will be giving her presentation to tell us about some of the approaches she's been using to study head and neck cancer, dissecting cholesterol biosynthesis pathways. And Jiyoung, thank you very much for joining us today, and I look forward to hearing about your work.
Thank you very much, Dr. Shuk, for your nice introductions. Today, I'd like to share one of my research projects to understand the novel cholesterol biosynthesis pathway in head and neck cancer. Head and neck cancer, or head and neck squamous cell carcinoma, is a group of cancer that happens in the mouth, sinus, nose, and throat areas, as you can see in these images. Then cancers can happen in the in between head and neck, including tongue, lips, and pharynx and larynx, and even the oral cavity. So based on the location of primary cancers, head and neck cancers can be subtyped, such as nasopharyngeal or pharyngeal head and neck squamous cell carcinoma. Head and neck cancer is one of the most common cancer, and as you can see in this plot, that head and neck cancer incidence is slightly but increasing over time in the United States, and we can see the annual increase, the number of new cases with head and neck cancer in the United States. So for head and neck cancer, smoking and drinking are known risk factors for this cancer. But interestingly, obesity is inversely associated with the risk of head and neck cancer. Also, high BMI of patients present better outcomes, particularly for the patient with chemotherapies. In general, obesity is not associated with head and neck cancer overall survival. But another interesting part of head and neck cancer is human papillomavirus associated head and neck cancer incidences increasing, particularly for young patients in Northern America. But HPV positive tumors, conversely, showing better responses to the chemotherapy or radiotherapies compared to the HPV negative tumors. Again, obesity is not associated with HPV status. In clinic, more problem is recurrent or metastatic head and neck cancer, because recurrent or metastatic head and neck cancer shows poor prognosis with a shorter survival rate, even with chemotherapy or systemic therapies such as targeted therapy and chemotherapies. To manage these kind of clinical problems, the more advanced therapies such as immune checkpoint inhibitors or epidermal growth factor receptor inhibitors such as EGFR inhibitors, has been applied to the patient to manage head and neck cancer. So basically, we had come up with this idea, like how do we understand head and neck cancer, particularly focused on metabolism. So we had a collaboration with medical oncologist Dr. Choro Kim in Aju University in Korea. So our previous postdoc performed RNA-seq analysis using patient tumors and then compared RNA-seq data with their paired normal tissues. So our patient cohort include all different types of subtypes, such as oropharynx, hypopharynx, and oral cavity, and tongue. And our patient cohort also has mixed tumors, HPV positive and negative, and also both female and male. So using this RNA-seq data, we basically look at the how different cancer cells compare to the normal cells with like head and neck cancer, so that we identify that cholesterol biosynthesis pathways and steroid biosynthesis pathways has been highly upregulated in cancers compared to normal tissues. And we could also see that many of the pathways involved in these functional alterations are mostly glycosylation. But we could also see that many other metabolic pathways, such as amino acid or pyruvate, TCA cycles, those metabolic pathways has been downregulated in cancer compared to the normal tissues. So we were more interested in these top pathways, steroid and cholesterol biosynthesis pathway in head and neck cancer. So gene set enrichment analysis shows there number of genes involved in these pathways. Then we could see that they are highly abundant because cholesterol further becomes a steroid. Then Burkina shows that fold differences with p-values indicating that these genes are highly or significantly overexpressed in cancer compared to the normal tissues. So I summarized these metabolic pathways in this diagram, showing that these genes involved in these pathways are upregulated in head and neck cancer. So this shows that de novo cholesterol biosynthesis pathway, starting from acetyl-CoA through mevalonic pathways, then squalene epoxide comes to the cholesterol through two different pathways. One is block pathway, the other one is KR pathways. So I simplified de novo biosynthesis pathways here, just focusing on the genes highly upregulated in our patient data set. But in general, this pathway has more than 30 steps, including a lot of energy requiring steps. So when we look at the cholesterol biosynthesis pathways has been increased in head and neck cancers compared to normal tissues, then this finding is not really surprising because other studies also show that cholesterol biosynthesis pathway genes are highly upregulated in many other types of cancers, as you can see in these figures. Many genes involved in these pathways are upregulated. At the same time, cholesterol exporters such as ABCA1 is also downregulated in many types of cancers, as shown in blue boxes, indicating that many cancers may need extra cholesterol by so that they are overexpressing or amplifying these metabolic genes in cancers. Because cholesterol is the a non-essential lipid, which is principal steroid required for their cell membrane synthesis. So this is a part of the membrane of the plasma membranes. So it is required for rapid cancer cell growth, so that we can easily think that cholesterol is more required for cancer cells for rapid cancer growth. Cholesterol is also an important precursor for the biosynthesis of steroid hormones, bile acid, and also vitamin D. Cancer cells maintain cholesterol homeostasis mainly two different ways. One is uptaking cholesterol from blood using the receptors, and the other way is making synthesizing cholesterol from acetyl-CoA, as I showed in my previous slide. So by maintaining cholesterol levels in head and neck cancers, so they synthesize or uptake. Because we've been seeing a lot of upregulations of genes involved in this pathway, so we hypothesized that head and neck cancer cells specifically need more cholesterol for their growth. That's why they are overexpressing these enzymes or genes. So first we tested head and neck cancer cell lines in the lab by providing extra cholesterol to the cell lines, and then we monitored cell growth and survival. So these growth of cells were monitored by measuring confluences. Then the extra cholesterol was provided at the time point with arrow indicate. Then we monitored the cell growth over time, but we did not see the growth differences by providing more cholesterol or not. And further, we measured or we quantified cell viabilities after treating cholesterol without LDL or long-term survivor by colony formations by providing cholesterol or cholesterol LDL. But we did not see any growth survivor differences by providing more cholesterol to the head and neck cancer cells. So because we provided one type of lipid, so we thought that what if we provide extra lipid, including cholesterol, with other types of lipids, such as fatty acid, triglyceride, to the head and neck cancer cells, then we monitored cell viabilities. However, we did not see any extra growth or more survivors of head and neck cancer cells with extra lipid. We saw a little bit of like toxicity with extra lipid to the cells, but we did not see any increases of cell growth. Then we further look at whether extra cholesterols are really utilized by head and neck cancer cells. So we stained cholesterol after treating cholesterol without LDL. Then we measured cholesterol levels per cells by Philippine staining. So you can see they're like white particles in these images indicating cholesterol lipid. Then after measuring cholesterol content after treating cholesterol, but we did not see the differences between cholesterol levels even after cholesterol LDL treatment. So we went back to the data set to test whether head and neck cancer cells also altering cholesterol transporters. Then we did not see the gene expression changes, indicating that head and neck cancer cells particularly do not take up blood or serum cholesterol to the cancer cells. Rather, they are probably making more cholesterol to support their cholesterol biosynthesis or cholesterol homeostasis. But as a proof of principle, we wonder whether cholesterol is important for head and neck cancer cells. So we basically used a small drug to get rid of cholesterol from the cells using methyl cyclodextrin, MCD. So MCD basically does deprive cholesterol from the cells. So when we treated head and neck cancer cells with MCD dose dependently, we could see their decreased cell growth. However, when we replenish cholesterol with MCD together, then we could see their cell growth was rescued from cell death. We further tested by providing extra lipid. Then we could see that extra lipid additions with MCD together could rescue cell death, indicating that head and neck cancer cells, of course, need cholesterol for their survival and growth. So to come up with the basic idea why head and neck cancer cells overexpressed all these blue genes that are involved in this cholesterol biosynthesis pathway. So we tried to interrupt this biosynthesis pathway by blocking the key rate-limiting enzyme HMGCR. So HMGCR is HMG-CoA reductase, a rate-limiting enzyme for these pathways. And we use the small drugs to inhibit HMGCR statins. The statin is known to reduce total cholesterol levels, but also it has been shown to reduce cancer cell survival or growth. So particularly simvastatin is the one of the types of drugs that has been shown to suppress cancer cell proliferation and migration. And then this is a summary of their findings. So we basically use this drug to test what if we interrupt this whole biosynthesis pathway, then what will happen? Then we treated head and neck cancer cells with statins, then we could see the significant suppression of cell viabilities with the statin treatment. So in other words, it says that head and neck cancer cells highly overexpressing these enzymes, then interruption of this pathway can be lethal to the head and neck cancer cell viabilities. So based on these findings, then we focused on the particularly two enzymes, DHCI7 and 24, that are highly upregulated in cancers compared to the normal tissues, but hasn't been much studied in cancer field for its functions, whereas other genes are well studied in cancer field. So DHCI7 and 24, as you can see in this figure, are involved in the last step of cholesterol biosynthesis. However, DHCI7 are also responsible for this pathway, these steps, and then DHCI24 is in general converting block pathway to KI pathway. So in other words, even though they are involved in the last step of cholesterol biosynthesis pathway, however, this whole pathway has been regulated by these two enzymes. As you can see, cholesterol biosynthesis pathway mostly happens in ER, so DHCI7 and 24 are ER membrane-bound proteins and involved in this cholesterol biosynthesis. But generally, it's not well known in cancer or cancer context. DHCI24, which is 24-dehydrocholesterol reductases, is also known to be overexpressed in adrenal gland cancer cells, but at the same time, it is known to be underexpressed in the tissues of Alzheimer's diseases. So we are more focused on these two genes, DHCI7 and 24, which are involved in the very last step of cholesterol biosynthesis pathway. So before going on to the data analysis, then we further validated whether DHCI7 and 24 that we found in RNA-seq data are also well overexpressed in patient tissues and cell lines. So first of all, we did QRT-PCR to see DHCI7 and 24 expression in the independent data set from the patient. Then we could see that both of them are significantly overexpressed. And immunohistochemistry assays also confirmed DHCI7 and 24 expressions are highly niched in the tumors compared to the normal tissues. We further validated DHCI24 expressions in tumor compared to normal tissues, showing that consistent overexpression of these two genes in tumors. So more important part is here because we had our postdoc had another data set for single cell RNA-seq, and then we identified that DHCI7 and 24 are exclusively higher in tumors compared to the other immune cells that are infiltrated into the tumors. So indicating that both genes are exclusively expressed in tumors, potentially suggesting that they are potentially good therapeutic targets. Then these DHCR7 or DHCR24 also predict patient outcomes. As you can see, when we analyzed TCGA data with head and neck cancer tumors, then we could see that patients with tumors that express high levels of DHCR7 show shorter survival, and also DHCR24 high expressing tumor showed shorter survivors. In general, when patients express high levels of, 7 and DHCR24, then their survival rates are significantly shorter compared to their counterpart controls that express low levels of DHCR7 and 24. So we came to this, the idea like, what if we block DHCR7 or 24 using siRNA, whether this is sufficient to stop cell growth or cell viabilities. Then when we used for different head and neck cancer cells and then silencing DHCR7 or 24 significantly suppressed the cell viabilities, and then SH stable cell line also suppressed cell viabilities and survival. Then we could see that like their morphological changes by knocking down DHCR7 and 24. Then we further like try to CRISPR knock out DHCR7 and 24. However, cells became like a lethal with this knockout. As you can see, CRISPR knockout completely wiped out proteins. However, their viability is really, really low, indicating that it's because DHCR7 and 24, particularly 24, is important for cholesterol biosynthesis, so converting desmosterol to cholesterol. So we thought that because knockout of DHCR24 need more cholesterol for rescuing their survivor. However, even providing cholesterol to the knockout cells did not rescue their survivor or growth. So their long-term survivor was very poor. So we could not use any CRISPR knockout cell lines for further experiment. So because we have small inhibitors to target DHCR and 24, so we, in parallel, we use the small inhibitors to inhibit DHCR7 and 24 to understand how important these genes in head and neck cancer cells. So to block DHCR7, we used AY9944, and for DHCR24, we used the trepanel. Trepanel is this is the first synthetic cholesterol-reducing drug in the United States in early 1960s. It is known to inhibit DHCR24. However, it's because of the side effect, so it became off market right away. But as a proof of principle, we used the trepanel to block DHCR24 in head and neck cancer cells in vitro. Dose-dependent treatment of trepanel decreased cell viabilities in all of the head and neck cancer cell lines that we tested in our lab. But when we use this drug, like certain concentrations, such as 10 micromolar that suppresses cell viabilities, did not change the total cholesterol levels in head and neck cancer cells, but still sufficiently suppressed the cell viabilities. At the same time, we use the AY9944, which is suppressing DHCR7, that we could see that dose-dependent suppression of cell viabilities. Again, DHCR7 converts 7-dehydrocholesterol to the cholesterol, and AY9944, suppresses DHCR7 activity. Because both drugs are blocking the last step of cholesterol biosynthesis pathway, so the reason why these two cells killing cancer cells is because of cholesterol reduction. So we provide the extra cholesterol to the cells with these either drugs to see whether cholesterol addition can rescue cell death from this drug treatment. However, as you can see in these two data, AY treatment with cholesterol or triple with cholesterol did not rescue the cell death. Also, cell viabilities also indicate that cholesterol addition with AY9944 or triple with cholesterol did not rescue the cell death or reduce the cell viabilities. So this data suggests that blocking DHCR7 or DHCR24 reduces cell viabilities. It's not because of cholesterol. We also combined AY9944 triple together. So what if we block two enzymes at the same time, whether we see synergistic effect, but we did not see the more additive effect when we combine AY9944 or triple in head and neck cancer cell lines, indicating that blocking either gene is sufficient to suppress cell growth or cell viabilities. And then this is a long-term survival by colon information assays that are treated with AY triple or AY triple at the same time without cholesterol rescues. Then we could see the consistent data indicating that blocking either enzymes is sufficient to suppress cell survival. So to further investigate what is happening in this metabolic pathway, how like metabolites are changed by blocking DHCR7 or DHCR24. So we first tested by looking at the metabolic changes by metabolomics analysis using DHCR24 silenced cell lines. So after silencing DHCR24, we could see the upregulated or increased desmosterol and the 7-dehydrocholesterol. However, we did not see the cholesterol changes or further upstream of this pathway. Lethosterol or lanosterols has not been changed by blocking DHCR24 for 48 hours. So we could see that like blocking DHCR24 accumulate desmosterol, and also it accumulates 7-DHC in the cells. So we could see that like enzyme substrate and product ratios are highly changed by increasing its ratios, desmosterol over cholesterol or 7-DHC to the cholesterol, indicating that these kind of imbalances in between substrate and the byproduct ratios can be toxic to the cells, not because of the total cholesterol reductions by knocking down DHCR7 or 24. So we further studied what if we provide extra substrates such as 7-dehydrocholesterol or desmosterol to the cells, then what is happening? Then we used the 4 different head and neck cancer cell lines again, and the cells were treated with dose-dependent 7-dehydrocholesterol without or with LDL. Then we could see the significant suppression of cell viabilities. But desmosterol with the same concentration without LDL did not change the cell viabilities at all, indicating that probably 7-DHC can be toxic to the cancer cells that can decrease cell viabilities. We further tested using cancer cells by providing LDL or cholesterol LDL or same amount of 7-dehydrocholesterol or desmosterols. Then we could see that cell viabilities are significantly suppressed when cells are treated with extra 7-dehydrocholesterol regardless of LDL presence. And then we further look at the cell survivors by colon formation assays using multiple cell lines. Then we could see that cholesterol, desmosterol without LDL doesn't change cell survivor or colon formations. However, extra 7-dehydrocholesterol without LDL decreased cell survival and colony formations, which is consistent with our viability assays. So far we had a conclusion that head and neck cancer cells are highly increasing the expression of genes involved in this novel cholesterol biosynthesis pathway, including DHCR7 and 24. So when we silence DHCR7 or 24 using siRNA or 2 small drugs, then we could see that cancer cells accumulating desmosterol and 7-dehydrocholesterol in the cells. And particularly, we think that 7-dehydrocholesterol accumulation might be causing cell death to decrease their viabilities. So to understand further, how extracellular or metabolic intermediate can cause cell death, we are more into the mechanistic understanding how these can kill the cells, which pathways the cell death mechanisms. So we basically look at the annexin 5 staining to see whether cells increase apoptosis after treating triparanol and also 7-dehydrocholesterol dose dependently. So this is undergoing. So some of the data are only 2 time point. However, our data indicate that when we block either DHCR7, DHCR24 using small drugs or providing extra 7-DHC could increase cellular apoptosis. We could also see the increased apoptosis of head and neck cancer cells with 7-DHC treatment. Then by looking at the cleaved PARP expressions, however, desmosterol treatment with the same concentration did not change cleaved PARP expressions, indicating that desmosterol is not really inducing cellular apoptosis, whereas 7-DHC is increasing apoptosis of the cells. So more importantly, when we provided 7-DHC or desmosterols to the head and neck cancer cells without LDL, then these substrates could be easily converted to extra cholesterol. So we look at the Philippine staining to measure whether when we provided extra substrate to the cells, which can kill the cells or either doesn't do any thing at all to the cells, but these 2 substrates were not utilized by cancer cells for further cholesterol biosynthesis. So we could see that extracellular or exogenous 7-DHC could increase cellular apoptosis. Then we wanted to know why or how this can happen. So we reasoned why, because the very far upstream of the cholesterol biosynthesis pathways is acetyl-CoA. So this acetyl-CoA is coming from mitochondrial respiration. So that once we block or once we interrupt the pathway in this cholesterol biosynthesis pathway, then it might alter acetyl-CoA production from mitochondria, which might also affect mitochondrial biogenesis. So we reasoned interruption of either DHCR7 or DHCR24 or exogenous 7-dehydrocholesterol addition to the cancer cells might stop mitochondrial respiration here, altering reactive oxygen species productions. So after knocking down DHCR24 using siRNA, then we measured mitochondrial ROS levels. However, as you can see in this data, we did not see any changes of mitochondrial ROS levels by knocking down DHCR24 or even rescue with cysteine additions to the cells, indicating that mitochondrial ROS is not the reason why interruption of these mitochondrial cholesterol biosynthesis interruptions causes cell death or apoptosis. So another cell death mechanism is ferroptosis, because previous studies also showed that that cholesterol biosynthesis pathway can regulate ferroptosis through inhibiting lipid peroxidations. So in the uh, acute lymphoid cells, accumulated squalene show their lipid peroxidation suppressions, which further protect the cells from the ferroptosis. So we hypothesize that increasing biosynthesis pathway for cholesterol is probably highly associated with these ferroptosis pathways or ferroptosis, in addition to the apoptosis. So we tested by treating ferroptosis inhibitors to the head and neck cancer cells, which doesn't do anything with the right range of the concentration that we treated. As you can see for the proliferations or also viabilities. Then we further combined ferroptosis inhibitor with AY9944, which is inhibiting DHCR7, or triparanol, which is inhibiting DHCR24. Then we did not see the rescue effect by adding ferroptosis inhibitors, indicating that blocking DHCR7 or 24 is causing cell death through apoptosis, not through ferroptosis. So we came back to the original idea that cholesterol biosynthesis pathways has been highly upregulated, and as you can see from the squalene epoxidates, then all the pathways happens in ER. So that we hypothesize that these overexpressed genes and these metabolites may regulate ER stresses. So in the ER, unfolded or misfolded proteins, overloaded proteins cause ER stresses through the three major pathways. So one is through PARC pathway or ATF6 or IRE1 pathways. So when ER is overloaded with these stress inducers, then turns on the signal transduction such as PARC phosphorylation, further EIF2 alpha phosphorylation that turns on ATF4 and the downstream effect in the nucleus. Then ATF6 is also cleaved based on the signal transduction, based on the stress signals, then further go into the nucleus to turn on the response genes. Then also similar way, IRE1 activate downstream signaling to respond to the ER stresses. So we tested whether overloaded 7-dehydrocholesterol in head and neck cancer cells might increase these ER stresses. So after screening these or the target, so we could see that 7-HC treatment may increase this phosphorylation IRE1 and also EIF2 alpha phosphorylation by 7-HC in dose-dependent manner in head and neck cancer cells, which is suggesting that increased ER stresses by these accumulated 7-HC may further activate cellular apoptosis. So far, our data suggests that head and neck cancer cells overexpress the Novo biosynthesis pathway for cholesterol in cytoplasm and in ER. Then particularly these two genes, DHCR7 and DHCR24, that are involved in the last step of cholesterol biosynthesis pathway, are particularly important to suppress ER stresses by maintaining low level of or homeostasis of desmosterol and 7-dehydrocholesterol levels. However, when we inhibit DHCR7 or 24 by a small inhibitors, then we could see there DHCR7 and 24 activities decreased, and then we could see that particularly 7-dehydrocolesterols accumulated in ER that further activate ER stresses or overloading ER stresses that can further increase cellular apoptosis. so that we could see that like just providing 7-DHC to the cells could cause cancer death. So summary of my talk today is we identified the novel cholesterol biosynthesis pathways is unregulated in head and neck cancer tumor tissues compared to the normal tissues. Then particularly we are focused on these two enzymes, DHCR7 and DHCR24, the last steps of this pathway. Because these two genes are highly overexpressed in cancer specifically, also predict poor outcomes of the patient survivors. So when we block these, either of these genes using siRNA or using small drugs, we could see the cancer cell viabilities decreased. And the reason is because we could see there like accumulated 7-DHC, which could further like induces the ER stresses and further cellular apoptosis. So we could get the idea that the why cancer cells increasing DHCR7 and DHCR24, or in general whole pathway, is to protect cancer cells from ER stresses by removing toxic intermediate accumulations such as 7-DHC in head and neck cancer cells. So this project is done by our previous postdoc Boksun Lee and master students Hing-Tian Liu and other lab members also involved in this project. And these are my collaborators for patient tissue and analysis by Dr. Choro Kim in the Ajun University School of Medicine, or our clinical collaborators here at MFA, and then Dr. Ivan Lin and Mano Chi is also collaborators for mass spec analysis for metabolomics, and these are my funding source. And this is all about my talk today, and I am happy to take any questions.
Thank you very much, Jiyoung. If there are any questions from audience members, feel free to unmute yourself and ask. Maybe I'll start with a question, giving people a chance to kind of formulate their thoughts and ideas. I found it really interesting that targeting cancer, I'm targeting cholesterol biosynthesis has such a profound effect on head and neck cancer. And the beauty of some of these virtual meetings are you can quickly pull up Google and do a quick search on things. It's interesting that statins are associated with improved cancer survival. But I know with statins being really far upstream, the cholesterol biosynthesis, there's going to be different metabolites that build up. Um, do you know, or would you suspect that the mechanism for statins improving outcomes, do you think it's going to be significantly different from the mechanism that you're studying here with your drugs targeting DHCR 24 and 27? Like, do you think the metabolites are still having a significantly different effect, or is it kind of still a similar cholesterol-based
Yeah, no,
phenotype?
that is really great question. So, of course, I looked at the literatures indicating is because statins, particularly statin, has shown anti-cancer activity so many cancer types, including breast cancer, liver cancers, but particularly their reasons is because the total cholesterol are reduced by statin treatment. However, based on my research, literature research, then head and neck cancer particularly do not correlate with the survivor or recurrences or risk of head and neck cancers. So in other words, statin users, people like study, like, analyze the data to see whether statins are better for head and neck cancer outcomes, but there is no correlation, particularly for the head and neck cancers. But in other types of cancer, as you can see here, are known to not just blocking mevalonic acid production, further like cholesterol productions, but in general, these statins are known to induce apoptosis and also other signaling, TNF-alpha, MIG, and everything. So it is known to affect many other targets in cancer in general to suppress cancer growth. So I assume that it's because my data here also showing that like statin treatment decreases cell viabilities, then I assume all those downstream after mevalonic acid are all suppressed by statin treatment. Then I thought that it's because all of them are required for their cancer cell viability and growth. That's why statin is killing cancer cells. But with this, I think we need more further like clarification is because I am testing after treating statins, then I assume every downstreams are all downregulated. But what if I add 7-DHC alone to the cancer cells? Then is that killing further of the cancer cells because 7-DHC is the causing ER stresses, or because it is rescuing the downstream effect? So my answer is like we need more investigation to answer your questions. And again, statin has no correlations with head and neck cancer outcomes.
Thank you. Bill?
Yeah. Can you hear me okay?
Yeah,
Yes.
sure.
Yes. When I was a postdoc at the Aging Institute, we were looking at the longevity of mitochondria cells. And I had one project where I decreased the vitamin E antioxidant in the animals. And within 3 months of that diet, they had the changes in the mitochondria, lipofuscin, that was seen in the 2-year-old rats. So, and you mentioned vitamin D, which has protective effects probably at the cell level. Is there any endpoint where you looked at the oxidative stress in the cells before and after depletion of this cholesterol?
So Bill, you asking me whether I looked at the further like cholesterol modified further steps such as vitamin D?
Yeah.
Good question. I am not showing here all those data. It's because of our metabolomics analysis also further like look at the like cholesterol, oxysterol, and further vitamin D. We did not see any changes by blocking DHCR24 in our cell lines. So we did not see this kind of further biosynthesis in our data.
Just a quick question. In recent years, there's a lot of emphasis on the cell exterior membrane lipid rafts, which have cholesterol in them. And if you're inhibiting the components of the lipid rafts, have you any data where some drug binding or some effect on the cancer drugs at that level of receptors is affected at all by the like a stress test of some sort against the cancer drug treatment?
That sounds really exciting. So yeah, I have no clue about it. So we can discuss more about if we have any sensors that can detect lipid like triglyceride or cholesterol like lipids with the sensors. That would be really interesting, but I have no idea.
A lot of elegant data, quite a lot of work.
Yeah, of course.
Thank you.
Sure, thank you.
Hi, Jiyoung. This is
Yeah.
Sabi here.
Hi, Sabi.
Really enjoyed your talk.
Thank you.
So as you know, what sounds like the cells that you used are using the cholesterol as possibly a fuel substrate. And do you think there's any interplay with glucose per se? And have you used the media that you used is, I was actually talking to Brett a few days back, that most of her experiments that we use for DMAM and so on are hyperglycemic, which automatically promotes cholesterol formation in other pathways that we don't need to go into that. But, how does glucose interplay with cholesterol utilization and cholesterol metabolism in cancer? This is also brings into the fact that there are so-called pathways that people have talked about in glucose and glucose utilization in cancer per se.
Mm-hmm. Mm-hmm. Yeah, that is really great question. First of all, for cholesterol biosynthesis, again, the far upstream is acetyl-CoA, which is coming from glucose and glycolysis pyruvate, and then coming into the TCA cycle and coming out acetyl-CoA. So this acetyl-CoA is from glucose per se.
Right.
So, and then many, as you mentioned, of course, all the cancer cells are in the hyperglycemic condition, which is way too high. Even though I'm saying that like head and neck cancer cell lines that I'm culturing here has only 5 millimolar glucose, but still this is not like low. So, I can say that like this glucose is coming from breaking pyruvate and providing acetyl-CoA for this cholesterol. So cholesterol is basically coming from the, I cannot say that, cholesterol biosynthesis basically use as acetyl-CoA from glucose. So they're like glucose levels affecting for sure these pathways. However, given the like conditions there, like when they provide sufficient acetyl-CoA, then their pathways is not just to support total cholesterol levels. Again, the cancer cells somehow don't like to accumulate some intermediate in this pathway. They are overexpressing these enzymes to pass their substrate to the product quickly. So the ER doesn't need to burden too much metabolite in ER to reduce ER stresses. So this is what I found here. So your question, can you remind me your question again?
Yeah, so, as you, I think, touched in the very beginning of your presentation, that the role of so-called de novo lipogenesis. And as you know, that is very dependent on glucose levels of the cell that is exposed to.
Okay.
And I was essentially trying to look into what was the glucose levels in the conditions that you looked at, and how would that change so-called de novo lipogenesis of your cells per se, and whether there is an interplay between glucose and cholesterol, not only formation, but also uptake by the cells per se. So, but I think you pointed out that, this is acetyl-CoA is a synthesis byproduct of glucose metabolism, which feeds into the utilization and TCA cycle production and so on.
Right.
But clearly there's a interplay between the hyperglycemic milieu and the cholesterol utilization and uptake by the cells. But,
I guess so. Yeah, so complicated in between like glycolysis and the oral glucose and how that affect cholesterol biosynthesis pathway. Yeah, I wish I knew all those like.
But what you said is you did all your experiments in so-called 5.5 millimolar, right?
Yeah, 5 millimolar glucose conditions. It's because all of the cancer these cell lines do not use DNEM. So they are quite like lower compared to the other types of cancer cell lines.
So the other point that you made is that when you expose the cells to simvastatin, now simvastatin, as you know, is a HMG-CoA reductase inhibitor, which works primarily at a hepatocyte and thereby prevents cholesterol formation at the hepatocyte. Would such an agent work similarly in other non-hepatocyte cells as well?
Yes.
Was my
yes
question.
Yeah. Right. There was the That's how like HMGCR inhibitors, like statins are applied to many cancers in even clinical trials because many people try this HMGCR blockade using statins, then showed similar effects such as reducing total cholesterol levels. Then cholesterol again is important for membrane biosynthesis or membrane synthesis. So just reducing total cholesterol levels in cancer cells by blocking HMGCR using statin was sufficient to suppress rapid cancer cell growth in many cancer types. That's why simvastatin has been in clinical trial. Then, as far as I know, the simvastatin with chemotherapy combination for breast cancer clinical trial has failed because there's no additive effect, although retrospective speculation or study whether statin users showing better outcomes with breast cancer. For example, they showed positive correlations, but
Right.
which was, I don't think it passed or it succeed in clinical trials. So it may need more. So when it comes to the patient, as you mentioned, statin of effect liver favor most effectively maybe, so that their total systemic regulation might not be as the same as when we tested single cancer cell lines or
Right.
animal models. So we need further like validation or investigation to understand how this interorgan and systemic regulation of statins and cancer later. But at this moment, statin is known to suppress total cholesterol levels that further affect cancer cell proliferation.
Just one more point, I'm sorry. As you know, that simvastatin works through cytochrome P4 pathway,
Yes,
and then it's essentially a lipophilic statin as compared to other statins such as rosuvastatin,
right,
which is hydrophilic and uses a different cytochrome P5.
Yes.
Do you think those differences make any difference, would make any difference as far as these experiments go?
That is a really good point. I used the simvastatin here in my studies. Then also I had a like same question like, oh, do I need to hydrophilic, hydrophobic, those kind of two different like types? Then I may try, but I have no answer at this moment. But this is a really good point.
Thank you. Enjoyed your talk.
Thank you. Is there any other questions?
Dr. Lee,
Yes.
I would like to compliment. It's really an informative and a nice presentation. Nice work you are doing. I would like to know just the nitty-gritty of the assay where you said that these two, the DHCR7 and DHCR24, does not work synergistically. Although here in this slide, I see that at least in one arm of the pathway, it is one after the other. I mean, DHCR7 is above DHCR24. So I was surprised, like, have you tried like putting one after the other, the inhibitor, or just together, or just reversing the thing, like putting one first and then the second first, like that? How about that?
Thanks, Richie, for your nice suggestion. As you can see, I really oversimplified because I was focusing on the genes overexpressed in our patient data. But as you can see, DHCR7 are involved in not just final steps, but other steps making desmosterol, which further utilized by DHCR24. Then DHCR24 is like converting everything like dihydroxide of 24 from block pathway to KI pathway. As you can see, lanosterol is converted to these lanosterols, and then everything is going into one direction. But again, these two enzymes are in were convoluted in this pathway. So just blocking one pathway was sufficient to see the effect in our preclinical data, so that we don't do much like sequential like suppression, after blocking DHCR7 and then later DHCR24 or vice versa, because we think that like just blocking one might be sufficient to see the phenotypic changes in our data set. But thank you for your suggestion.
Sure. Thank you.
Great. Well, Jiyoung, on behalf of myself and my co-organizer, Dr. Lee Frame, thank you very much for your contribution to this seminar series. It was a great presentation and there's a nice lively discussion afterwards. So thank you very much and best of luck to you with this research.
Thank you so much, Dr. Shook.
Sure. Bye.